Hello and welcome back. Today we are tackling evidence for evolution. So we're going to look at the specific science between comparing organisms, seeing if they have common ancestry, and how we can actually tell how organisms have evolved over the years. So let's dive into it. Before we really get started, I want you to note the four major categories of evidence that we're going to be talking about today. The first is the fossil records. This is both what we have found and what we are still currently finding out as we explore areas of the Earth. Body structure. This has a number of parts within it, but the idea of how organisms look and how they are formed with their bones. Similarities in embryology. So this is looking at embryo development. Embryo is like a stage in fetal development, and then the biochemistry, which is really like DNA. So let's dive into fossils right off the bat. With fossils, there are two different ways that we look at fossil records: absolute dating and relative dating. You can probably figure out which one is more reliable based strictly on their name. Absolute, I mean, that seems pretty profound, right? I'm absolutely sure that I know when this fossil was. And then relative dating is like, I'm relatively sure that I know where this fossil is. And that's pretty much what it means. So absolute dating determines the exact age of a rock. And this is done through looking at the radioactive decay of certain elements. So we will, take a sample of soil from somewhere and look at usually carbon, but there are other elements that we can look at and determine what their radioactivity is. And once we figure that part out, we can work backwards. So we do this by determining the half-lives, like how many half-lives an element has gone through. And you kind of just work backwards to determine when that fossil died, basically, because they would have had their full element, right? The full carbon when they were alive, but then as time goes on, they go through half-life after half-life, and basically it's exactly what it sounds like. Half of the carbon would be removed per half-life, and then the next time the half-life happens, half of the remaining carbon is removed, and it continues on until there's just barely any left. But you can still date the age of the fossil pretty much exactly, right? These are like millions of years ago, and we're talking like plus or minus 10. That's like not that bad. That's pretty close. And then relative dating determines the order of events something happens, right? Like I know that the older a fossil is, the deeper into the earth it's going to be, and the more recent a fossil will be, the closer to the surface it will be. But I couldn't tell you like exactly how many millions of years ago this organism walked the earth. I can't tell you all those specifics, but I can give you a general range of time that these fossils existed. Okay, we're going to talk a lot more about absolute dating in class because that's more reliable, right? That's a better way to look at fossils, but I just want to give you like a general sense of the differences. Let's get into body structures now. So body structures are pretty straightforward. If an organism is more similar to another organism in their body structures, then they're more likely to have a recent common ancestor. And I think that makes sense, right? If something is structured the same way as something else, then likely they came from the same source. And that makes sense. So there's three different structures that we're going to look at: homologous, analogous, and vestigial. I know these words are like wild, right? But we'll practice. So a homologous structure, we've used this prefix before, right? That means same. And homologous chromosomes, we've talked about that before. That means that they're the same shape, same genes on them, same size, same everything, right? So homologous means same. Structure, same structure. And what do you know? That's the beginning of our real definition of this. So a homologous structure has the same structure, but a different function. And you can see this in this first picture. All of these arms, right? A human, a cat, a whale, and a bat. These colored pieces, they're the same bones, okay? They're shaped slightly differently, but the bones themselves are the same, right? We have our phalanges, which are our fingers. Well, cats have fingers, whales have fingers, bats have fingers. And as you work backwards, these bones exist in all of these other organisms as well. So same structure found in all of them, but a different function, right? Our arms are used differently than a whale's fin might be used, but the bones are the same. So same structure, different function. What this shows is common ancestry. So somewhere, down the line, could be thousands, could be millions of years ago, there was a common ancestor between these four organisms. For analogous structures, it's the opposite of homologous. So different structure, but same function. And you can see this in like bird wings and butterfly wings, right? The wings obviously do the same function, right? They make that organism fly, but they're not the same structure. Birds may have bones in them, bats have all of these specific bones in them, but butterfly wings are just like a membranous, like flap. They don't have all of the bones in them, so although that they do the same thing, they are not showing common ancestry because they have a totally different structure. And then vestigial structure is this idea that we have structures that are no longer in use. We don't really need them anymore, but they still exist in the organism itself, which shows evidence of ancestry. Like whales have a pelvis bone because they used to have legs, but now they don't. And so they still have the pelvis, which would lead to legs, right? But it's just a structure that's no longer used. It exists, but it's not used. All right, getting into our third point is embryology. So embryo, as we talked about earlier, is the very early developing organism. And so if there are more similar structures in an embryo, then again, you're more likely to have a common ancestor. And all vertebrates, which is what this picture is showing, vertebrates, organisms that have a backbone, that's your vertebra, right? We're called vertebrates, have a common ancestor, and you can see that based on this chart of embryology. So this first row right across the top, they all look pretty similar. I would not be able to tell what these organisms are going to evolve in or develop into just by looking at this first row, because they all look super, super similar, right? There's a tail for all of them. There are gill slits. This is like where your pharynx, when you're developing, you have gill slits, but it turns into your pharynx. Okay, you see that on all of them. They all have these gill slits. But then as you continue to develop, you start to get your specific traits that you would see in these organisms, and then by the end, when you're like almost like a full-blown fetus, you really see the differences that are distinct to each organism, okay? But we are looking at, when we're talking about embryology, we're looking at this very first bit, very early development of organisms, okay? And last, but certainly not least, the biochemistry evidence. And this is talking about DNA. So I'm sure that at this point you have figured out the trend. If there are more similarities, in this case in the DNA or the amino acids, then they're more likely to be related with a common ancestor, right? The more similarities, the more likely to be related. That's like for all of this evidence that we've talked about. So this is nothing new. Hopefully you're familiar enough with this at this point to kind of put that piece together. Now there are two ways that you can look at this, right? You can look at biochemistry evidence via the DNA or the proteins. And I've given you an example of both of them. And we're going to practice this in class, of course, but I want you to just see this in the notes. So when we're talking about DNA, you are comparing specific nucleotides and determining which one has the most similarities. So in this example, I have an unknown species, and I want to see if it's more closely related to species 1, 2, 3, or 4. And so you just compare all of them to this unknown. So Our first three are ACT. Okay, well, the species 1 is ACA, so that's different. But species 2 is ACT, and species 4 is ACT. So then you just continue on trying to find the most similarities, okay? You continue on until you have figured out exactly which one has the most similar nucleotide sequence. That's all you want to do. In this case, our most similar species is species 4. And you can see that because there's actually only one difference in here, this G instead of a C. But that's basically all you do. You want to compare one species to the unknown at a time, that way you don't get confused. Or you could do it via the sequence of amino acids, and it's the same concept. You're just looking at the amino acids instead of the nucleotide basis. So again, you're just comparing which one would be the most similar, right? If we're comparing to humans, which one is the most similar? Well, I see lysine. Okay, we're looking at gorilla or chimpanzee here, and then glutamine. Okay, histamine. Okay, and then isoleucine. Okay, well, that knocks out gorilla, which means chimpanzee has the most similarities to the human. Okay? We're going to practice all of this in class, so don't worry if this is a little bit too much right now. We're going to make sure that you guys are absolute pros at looking at the evidence of evolution. All right? Proud of you for sticking through it, and I'll see you in class.